Hi class, so this video I'm going to go over action potential and contraction of a muscle, okay, and what kind of starts that process off, okay. So first we get some sort of signal, right? So we, tell our brain that we want to raise our hand to ask a question in class, okay. So what happens first is sodium flows into the neuron. Okay, this makes the inside of the neuron more positive compared to outside. Okay, and that positive charge or action potential of the neuron flows down to the neuron end bulb. Okay, so to the axon terminals and then to the end bulb. Now this end bulb here is communicating with this muscle. So this area here is known as the neuromuscular junction. Okay, so it's the site where the neuron meets the muscle. A junction is like where two things meet. Okay, so neuromuscular junction. So once we reach positive 30 millivolts within this neuron here, that's when these calcium channels here are going to open up. Okay, when they open up, calcium is going to flood into the end bulb. Okay, now what calcium does is we have, let's back up a little bit. We have a neurotransmitter known as acetylcholine, and it's found within vesicles. Okay, so we have acetylcholine, ACH, in our vesicles here at our end bulb of our neuron. Okay, so calcium floods in, and what this does, we have what's known as V snares and T snares. You don't really need to know this part, but they kind of anchor and they kind of twist together. So calcium causes them to twist together, and the vesicle gets pulled to the end of the neuron end bulb. So now I'm gonna erase this here. Okay, and now our acetylcholine can leave the synaptic end bulb and can be found in the synapse area. Synapse is just a space, okay, in this case, between the neuron end bulb and the muscle. Okay. So now we have acetylcholine released into the synaptic cleft. Okay. So once this happens, now potassium gates can open on the synaptic end bulb. So I'll draw my here. And now potassium can leave the cell, bringing us back down to negative 70 millivolts. Okay, so now we're at repolarization. Now the neuron is back at rest. No more calcium is coming in. Okay, no more calcium is coming in here. So now this area is closed again. Okay, just to show you what's happening so that this doesn't keep happening, right? So we don't just continuously let acetylcholine out. So, okay, now back here, acetylcholine is released to the synapse. Okay, now these little folds here are known as postjunctional folds. They're on the muscle fiber. Okay, so now on these folds we have ligand gated ion channels. So these are the channels that I showed you were like this. So they're shaped something like this. We have an inactivation gate and then inactivation gate and then an inactivation gate. Oops. Okay. So these are found in this fold. So let's draw one. We'll just draw one for an example. Okay. All right. So acetylcholine can bind to this channel here. Okay, so let's say our acetylcholine is this little pink dot. Whoops. Okay, so now acetylcholine has bound there. Okay, and now this channel is going to open up. So at first this is at negative 90 millivolts. Okay, so that's like its resting membrane potential of the muscle. Okay, remember negative 70 was of the neuron, negative 90 is of the muscle. Okay, and these are, this resting membrane potential is kept pretty constant due to sodium-potassium pumps, where 3 sodium are pumped into the cell and 2 potassium are pumped out. Okay, and then there's also potassium leak channels where potassium leaks out, and that keeps it more negative inside at about negative 90 millivolts inside of the muscle cell. Okay, so just telling you kind of how that stays as is. Okay, so after acetylcholine binds, I'm sorry, it's not this gate yet. It's just a regular, sorry about that. It's just a regular ligand gated channel. So acetylcholine binds and this channel opens up. Okay. So sodium is at a higher concentration outside of the cell. So now sodium can enter the cell. Now it can go into the muscle cell here. Okay. And potassium at the same time can move out. Okay. But there's more sodium moving in than potassium, so it's going to become less negative. Okay, so as sodium is moving in, we're moving toward our membrane potential, also known as the end plate potential, of negative 55 millivolts. Call this the end plate potential. Okay, so because of this sodium, we're getting closer to that end plate potential of negative 55. Okay. Okay, so now this gate here, we also have these channels. Okay, so let me make another spot here. Okay. Let me just draw this out with its activation gate and little ligand. Okay. So now our activation gate can open, so the swings open. Okay, and let me, or actually, let's just undo that and erase that. Okay, so now it's open. Oops. Okay, so now that gate is open and even more sodium can enter into this muscle cell. Okay, so now once this ligand gated channel opens up and more sodium rushes into the cell, now we're at our depolarization potential. Okay, so this is where action potential is going to occur. So this is plus 30 millivolts. Okay, so now this positive charge moves along down the T-tubule. Remember, these are the invaginations within the muscle fiber. And on either side of the T-tubules, we have the sarcomere. Now remember the sarcomere, let's draw our calcium in blue. So the sarcomere has lots of calcium. These will be our calcium ions. Okay, so the sarcomere just has lots of calcium. It holds it until it's ready to be opened up. Now, plugging up the calcium, we have these 2 proteins. You don't really need to know much about them, but I'll just draw them out here. And then we have these receptors. So, something like that. Okay, and then they're going to be on either side here. So once we reach that positive 30 millivolt action potential down our T-tubules, okay, behind it now these ligand gated channels are inactivating, so this little bulb here just kind of instead plugs up this area, and now sodium can't flow through anymore. Okay, so now it's going back down to its resting membrane potential of -90. But as this action potential is moving down the T tubule here, these two proteins come apart. Okay, so I'll just kind of unmake these here. Okay, and now we have these little openings where those proteins used to be. Okay, so now our calcium can rush out of the sarcoplasmic reticulum. And same thing happens on the other side with the other sarcomere. Okay, so now calcium can rush out. Now it's within the muscle cell. Okay, and now it can bind to troponin on the actin thin filament, cause a conformational change, which is a shape change. Tropomyosin will move out of the way, and now the myosin binding sites on the actin filament are available for myosin to bind and pull the thin filament in, okay, during a contraction. So now a muscle contraction can happen. Okay, so that's the basics of how the action potential occurs for a muscle contraction.